It's a pleasure and an honor for me too to be here and to be able to talk also like Rolf about infrastructure for life sciences. My talk will be about, I will tell you a little bit about some of the challenges we have in bioinformatics, but currently my two major challenges are first that Europe has a population of 750 million people. Switzerland is a small country of 8 million people. So how can I talk about the Swiss Institute of Bioinformatics after a talk about the European Bioinformatics Institute? This is my first challenge. The second challenge, it's the last talk of this workshop, and we are all fairly tired. So that's another challenge. So I decided to do it, to do a little bit of animation so that you don't completely fall asleep. So I will tell you about some of our challenges, some new challenges, some old challenges. So I will also give you a little bit of historical perspective. First, data science. Big data is, as you know, the fourth paradigm of science after empirical science theory, where people would go from experiments to rules. Then we had computation and simulation, where from rules you infer data, and now again we look at data and try to infer rules. So this is a new paradigm in the 21st century. But data science and big data existed for a long time. You all know very old data scientist. He's called data scientist, and he has been dealing with big data since for many years, namely with his good friend Big Datix. And these two, data scientists and big datix, they got along very well for many years. I can talk only about what we do in Switzerland. So for many years, we have been developing bioinformatics tools using data science to applied on data to develop a number of resources. Here you have a few examples. More than 30 years ago, we developed software to analyze data from life science called Melanie. Then in 1986, my colleague Amos Barock started a database. So that's nearly 30 years ago. Today, it is a collaboration with the EBIN group in the United States. I will just, and there are a few other examples, I will just spend 1 minute on this database, SwissProt, now part of the UniProt consortium. And this is a large encyclopedia of protein sequences. It's a manually curated, manually annotated database. It contains more than 500,000 annotated proteins, more than 200,000 literature references, and we get about 800,000 requests per month. And to do this, we have a team of 50 biocurators who spend their life reading articles and extracting, literally they are reading articles all day long and extracting the knowledge to put that into the database. And this is what we call, and Rolf has given you other examples, this is what we call infrastructure, the bioinformatics infrastructure for life science. And by infrastructure, it's mainly people, a little bit of machine, but it's mostly people. Machines are consumables. And this is what William calls the highway of life science research. And to do that, we need financial resources to pay the 50 people, and there are as many people at EBI. So this started in 1986. It worked quite well until we had a first challenge because of funding agencies who decided that, no, he doesn't want to pay for this highway. And that was 1996. Several media reported on that, including Nature and Science. And to solve that problem, what we did this was a universal problem, actually. There has never been funding for core bioinformatics resources, real funding for this infrastructure, for the highways. And so that is an old challenge. That was our challenge number one. What we did in Switzerland to solve the problem is that we tried to resist to funding agencies, and we created the SIB, Swiss Institute of Bioinformatics, first in Geneva and Lausanne, and then other cities joined. And today we are an institution federating bioinformatics groups from 16 universities and research institutes in Switzerland, including the two Federal Institutes of Technologies, ETH and EPFL. This represents about 700 bioinformaticians. And we are a very small country, but we are proud because we have the highest concentration of bioinformaticians worldwide, namely 88 bioinformaticians per million inhabitants. Our mission is to lead and coordinate bioinformatics in Switzerland and to join efforts throughout the country to advance biological, biomedical research and health. And we do that in two ways. First, by developing, maintaining, and providing bioinformatics resources, such as databases and software, both to the national and the international community. And we also federate the researchers in Switzerland, and we provide training. So that was 1998. So in 2002, we thought we had solved the fight against funding agencies, and we had solved the funding issue by creating this UniProt consortium. And since 2002, the SwissProt database, as part of UniProt, has been funded by these three large countries. So it's the European Union, United States of America, and Switzerland. 2002. 2008, another challenge. It's the omics data flood that we have been talking about since yesterday. It's in 2008 that the cost of sequencing dropped so much, and as a consequence, many, many more labs started to buy sequencers and produce data with sequencers that produced more and more data. And now we are flooded with data. This is the data explosion. Big datax became huge datax. Second challenge. 1 year later, what we thought was solved came back, our old challenge, and the EU, for a number of reasons that I can explain, including that now we have Elixir, Wolf talked about, decided to stop fund SwissProt, and since 2009, this database, which is basically used by all life scientists in the world, is only funded by the United States and Switzerland. And this is something which is going on. It's the main question. It has been published quite often. It's the question of how to sustain the digital infrastructure, how to sustain the data infrastructure for life science. So many people publish about that, but no funding model has yet been found. We have another challenge, more recent, is that bioinformatics and big data find applications in many different types of application fields. So I will give you 3 examples to be a little bit more concrete of activities of developments that have taken place in our institute. The first example is in the field of epidemiological surveillance, and this is something that was developed in the VitalAT group in Lausanne. It's a collaboration with the FAO, the Food and Agriculture Organization of the United Nations. The FAO is doing epidemiological surveillance of influenza outbreaks. So they have a database since the year 2000 containing epidemiological data on transboundary animal disease events. And it includes influenza, and this allows them to do outbreak surveillance. They have only epidemiological data. They have no molecular data. On our side, we developed a few years ago influenza database containing genetic and protein sequence data. It's a curated database based on public data. It has been now 7 years in production. And so as a collaboration with the FAO, it was decided to try to link these two databases. It was not an obvious problem, but working with the FAO experts, we designed an algorithm to match the data, epidemiological data, with molecular data. And this is now in operation at the FAO. And as an example of its usefulness, of its impact, when it was first used in 2013, it was tested on a recent influenza H7N3 outbreak in Mexico, in Jalisco. And this collaboration, this produces daily maps with the virus genetic landscape. And this is the one which came out in 2013, where you see that there are 3 clusters of viruses. One is South American, one is North American, and one is Eurasian. And if you look closer into the North American one, then you see the virus which is at the origin of the recent outbreak that happened in Mexico. And this allows to see that it's very close to 2 other viruses, one from chicken, one from human. So it allowed the FAO to find the origin, to get information about the origin of this new virus. This is the first example. The second example is in a completely different field. It's in archaeology. It's a development made by my colleague Christian von Mering and his group in Zurich, at the University of Zurich and the SIB. Together, it's a collaboration, also a quite large international collaboration. And they have been working on skeletons coming from Germany, about 1,000-year-old. And more precisely, they have been analyzing the dental plaque on the teeth of skulls 1,000 years old. And to make a long story short, they did both genomic DNA analysis and proteomic analysis, and they found quite a number of things. For example, they could do the complete coverage of bacteria, Tonarella forsythia, which is a pathogen known to be at the origin of gingivitis. The only difference with the known genome is that there are 2 areas which were not covered, and they think that what they have found is an early version of this pathogen which has evolved since. And this allows to see that the pathogens that we have to fight today were the same at that time. Another thing they found, they also sequenced and identified the genome of brassica, which is a type of cabbage. So we know that our ancestors were eating cabbage. We also, and they found that on the dead body, so they don't know whether the person that was analyzed was killed while he or she was eating, or whether she didn't brush her teeth. But it gives us interesting life science information about our ancestors. And the third example might look strange. In a bioinformatics institute, we have one group who is using, it's at the SIB, using the methodologies that we have developed for life science to apply them to digital humanities. And this group is currently working on analyzing Arabic versions of New Testament. This is also big data heritage. There are about 6,000 different Greek manuscripts and even more in Asian languages, and they are working now on an Arabic text which is here at the library at the Vatican to curate it and make it publicly available. So these are quickly 3 example. Our next big challenge for big data and bioinformatics, and we have been talking about that these 2 days, is personalized health, also known as personalized medicine or precision medicine. The goal of personalized health is to use different kind of knowledge, including genomic knowledge, but not only genomic knowledge, but also proteomic knowledge, other omics knowledge, and data from the patients, from the electronic health records, to predict disease susceptibility, disease prognosis, and treatment response, so to improve the treatment, but in general, the health of a person. A typical example is the use of specific tumor markers, such as genetic variation, to guide the therapy for lung cancer. And we've heard something about lung cancer today, and here is the quite typical example. In lung cancer, chemotherapy has an overall response rate of about 15%, and you see the same mutations that our colleague mentioned today. But it is known now that a patient who has an EGFR mutation, if you treat that person with erlotinib, that person will have instead of 15% response rate, will have a response rate of 70%. Similarly, if you treat with crizotinib a patient who has an ALK rearrangement, that person will have the overall response rate will be 80%, and the same for vemurafenib and the BRAF V600E mutation. Here, in this case, in this last case, it's even more interesting, because if you treat these patients with that drug, then you have a 40% response rate. But if you treat a patient that does not have this mutation with this treatment, then the cancer increases. So the situation gets worse. So this is our next challenge. We have already been working on personalized health at the SIB. That will be my last practical example. Again, in the VitalIT group, they have developed, in collaboration with 2 companies, a non-invasive prenatal test to detect trisomies. So today, until recently, the main way to test trisomies was amniocentesis, which has a risk of 1% to 0.5 to 1% to kill the fetus. In this case, the way it works, you just take blood from the mother, because we know now that we all have circulating DNA in our blood, and it was shown that in the blood of the pregnant woman, you find DNA from the mother, but also DNA from the fetus. Then you sequence it, and you just have to sort DNA out and look at the fetus DNA, and these are the results. So you see, for chromosome 21, this is something which was known before, but it was working only on chromosome 21, and the algorithm which was developed in Switzerland works with chromosome 18, 13, 22, and other chromosomal abnormalities. And this test that we developed is now on the market since 2013. So bioinformatics was mainly used in research, as bioinformaticians were developing tools and software and databases that were used mainly by life science researchers. But now our next challenge will be personalized health. And there is a new initiative in Switzerland, which is called the Swiss Personalized Health Initiative, whose goal is to create a national infrastructure allowing to share patient data to do research in personalized health. And the goal is to integrate universities, university hospitals, and also research institutes. And there will be 2 large clusters, one in the northern part of Switzerland, one in the southern part, including all the university hospitals. And the SIB, Swiss Institute of Bioinformatics, will have the role to do the data coordination part, which is a major challenge, because it means we have to make all these heterogeneous data interoperable. And they are data from quite different types, from hospitals, omics data, personal data. In addition, 80% of patient data are unstructured. And if you know that in Switzerland, it's a small country, but it's divided in 23 states, each hospital uses a different electronic health record system and different standards. So this is a real challenge, and this is supposed to start next year. We'll see how it is working. So as a summary, I quickly mentioned 5 challenges that we have had, some for a long time, some are very new: personalized health and data interoperability. I didn't talk about, but I have to mention the challenge of training and capacity building, because with all these new technologies, we have to train people, starting with physicians. And of course, we mentioned that also the ethical, legal, and societal issues, which are huge. There are so many questions which will come up. So as a conclusion, data scientists has a bright future, because we'll have a lot of work to do. It's today an essential part of life science research. It's also an essential part, and will be more and more essential part, of personalized health and the analysis of big data. But our old challenges are still there. We still have the problem of finding a way to fund these highways for life science. This is a very recent article which was published, amongst others, it was 2 weeks ago or 10 days ago, was published among others by Phil Bourne at the NIH, the head of BD2K, highlighting this problem of how can we sustain the big data ecosystem. So I thank you for your attention.